All right, so of all the things in orientation that I've been looking forward to the most, it's pretty much everything we're starting now and running through this evening. So you are about to hear a panel that is comprised of our scholars from cohorts 1 and 2. And it is very heartwarming, I guess is the word, for me to see them walk in to this building and come ready to present to you their research and their pathway that they've taken over the past few years. And thinking ahead that all of you will be on this same pathway. Not the same, but very similar pathways. And it's kind of remarkable. So what I've asked them to do today, I just want to let you know, we actually gave them some talking points. And as you'll see, they all sort of focused in different areas. But essentially what we wanted them to do was to share a bit about their journey as Exito scholars with you. So we asked them to give a really brief overview of their research. We asked them to tell a little bit about their experience being supported in their lab, or maybe even touch on any challenges they've had in their research environment. Things they wish they would have known. And I actually put would in all caps to emphasize, like, what do you really wish you would have known? They're going to try to give you some of that wisdom. And then any other tips that they could share with you. And like I said, they focused in different areas. So after they all give their presentations, I'll facilitate some question and answer, and then we'll open it up to the big group. And I really encourage you to use this opportunity to just pick their brains. Ask them whatever it is that you want to know about their role as Exito scholars. I guess you can ask them anything that you want to know, but I think they'll be best suited to tell you about their Exito pathway. And with that, is there anything else I should mention? No. I guess the only thing I will say is, just like with the research presentations, we asked them to hold this to 5 minutes, which is no small task, because there's actually a lot to share. So if you feel like there was more you want to know, just write that question down, and we'll definitely get a chance to cover it in the open Q&A. All right, so I'll go ahead and introduce them all, and then they'll all tell you more about themselves during their presentation. So all the way down here, we have Kim Dolan. Kim was one of our first scholars in cohort 1 and came to us from PCC, but when you got into Exito, you transferred here right away, right? Excellent. And then we have Sarah Florick. Sarah was in last year's cohort, and she's a PSU scholar. And then we have Sean Van Wahlgren. He came to us from Clackamas and was a cohort 1 scholar. Amazing. And here we have Diana Demchenko. Diana, you came from Clackamas too, yes? A little bit, but you were a freshman here. Okay, so you applied through PSU. And she joined us last year. And then we have Dejanique Brown. Dejanique came from PCC Cascade, yeah? And she was a cohort 1 scholar, as was Joseph Ramirez, who also came from PCC Cascade. Yeah, you came together, yes. I was going to say, it's very rare that I see you apart. Yes. All right, so I'll step out of the way, and I'll let Kim take it away.
Okay. Hi, everyone. So as she said, I'm Kim Dolan. I just finished my time as an Exito scholar, which is very sad. But I was working in the RICAL lab. I'm a biochemistry major, so I'm going to talk a little bit about my research and then my experience in that lab. So the RICAL lab is primarily focused on understanding the roles that membrane proteins play in biology and disease, and my project in particular was focused on this protein complex pictured right here, which is a member of the aquaporin family of water channels that's found only in the mammalian islands, and it's 2 regulatory proteins, which are these calmodulin proteins in blue on the bottom here. And so the focus of my research is on understanding how calmodulin is able to allosterically regulate water flow through the aquaporin 0 water channel. And in order to address this question, we will use a number of biophysical and structural techniques. And so kind of like the main overarching goal for any work that comes out of the RICAL lab is to obtain high-resolution structural information using this tool, which is cryo-electron microscopy, which is basically a high-powered microscope that uses an electron beam as its light source. And due to recent advances in camera technology and electron detection for this method, it's able to directly visualize biological macromolecules such as the aquaporin 0, chemato, complex. So as far as support goes in my lab, I've been very fortunate. The RICAL lab is a fairly newly established lab here at PSU, and as such, I have a lot of access to my PI, which is really great. So we do weekly meetings. Each week, someone will present their research, and then we'll kind of have this open session where we can ask questions or kind of critique or give them advice based on anything that they're dealing with. And then in this lab, and I think this will be the case for all of you, you're going to be paired with someone. So I work with a graduate student in the RICAL lab, and my project is sort of a corollary of his graduate project. So I have a person that I'm working in concert with, and so I'll always have access to him. As far as expectations go, it's really much like any job I've ever had. Be punctual, and then work hard. And I think it's important to, when you start, you don't know anything that's really hard. Don't be afraid to ask questions, especially in science. I think everybody has questions. You're always going to have questions. So definitely you have access to these people. They know a lot. Pick their brains, and they're all happy to share. And then also, you're going to make mistakes and get weird results, and that's something that you just need to talk through, and so just being honest about mistakes and that kind of thing. And then as far as what I would say is the thing that, I suppose I knew this, but something that I have to continually remind myself is it's very hard to start over in something that you know nothing about, and I think also the nature of research is that even if you're very good at it, you're going to have a lot of projects that fail, experiments don't work for whatever reason, even if they have been working. It's very easy to get down on yourself throughout the whole process, and especially in the beginning when if you're like me, you're going to be screwing up pretty much all the time. But that's very normal, and even great scientists struggle if some things don't work for them. So just continually remind yourself that not having success in your first month in the lab does not mean that you can never be a scientist. And maybe some of you guys know that, but if you don't, just, a little mantra for you. Don't be too hard on yourself. And then as far as tips for being successful in the lab, I think that, as I said before, asking questions is really huge. You want to really make sure you understand why you're doing something and not just simple methods. It will help you tremendously going forward. And then I think maybe the most important habits you can build in your beginning time as a researcher that will serve you throughout your career is being an excellent note taker. And this is for a number of reasons. One, just because you're going to need detailed notes once you actually start working on a real project. And then also, early on, you're going to have a lot of information thrown at you, and your PI and graduate students that you're paired with are going to be busy, so they won't repeat themselves. That's my time, so thank you.
All right, so everyone on your tables, I put printouts of my slides. I like to be able to have access to different ways to access information, and so I provided a physical way and a visual way, and I'm going to talk now. My name is Sarah. I do research at OHSU with a group that studies heart failure. We do a couple different things. We look at compounds to help reverse heart failure, and I'm looking at a patient population that got really advanced heart failure, and we're looking at different ways to support patients at the end of functioning hearts, pretty much. So my path to OHSU and research in general was really nonlinear. I started in a research group, as you guys know, I talked about earlier, really struggled, left that group, started with this group, I'm also a McNair Scholar, so I'm in the process of finishing my summer research project and writing a publication for August. And then I will continue with this group through Exito. This lab actually became an Exito lab because I asked them to. And that's something that you guys should be aware of. If there's a group that you're not quite sure fits your wants or desires, there is an option to talk to a PI and say, hey, will you come on to Exito and be a lab and be an RLC? So that's something to remember is if you don't have a fit, find something. Because I have found that if you don't ask, you'll never know. This is my group. So my PI is Dr. Habecker. Dr. Christopher Lee has collected all the patient samples that I use for my research, and so we always recognize him. Quinn Denfeld, I train with on the bench. She's helped me with developing my methods and other scientific questions. And then Bill is one of our chemists in our lab that's helping me with catecholamine work. So heart failure is super prevalent. We have a huge number of people suffering in the United States and worldwide from heart failure. A lot of the statistics and details of my work, because it's really hard to present in 5 minutes, is on the printouts that I provided for you guys. The symptoms can range really widely from just rapid heart rate to congestion and trouble passing fluids through the tissues. You end up with a lot of fatigue, and quality of life is significantly decreased with this disorder. It's a progressive disorder, and so as you get worse, you end up hospitalized more frequently, and the condition, there's no way to stop it. Once it has started, it just continues to progress further and further. So we look specifically at, I'm looking specifically at a biomarker. It's called ST2. It's, again, in the sheet. That is right here. And it is expressed after a myocardial infarction or damage to the heart. And it is progressively expressed more and more as damage gets worse. So when you have hypertrophy and fibrosis of the heart, you have higher expression. And there's a variety of different biomarkers that can indicate diagnostic and prognostic development of heart failure, and my biomarker specifically is prognostic, so it can't tell us, do you have heart failure? It tells you how bad is your heart failure. So I'm looking at a patient population who received these surgical implantation of these devices. And LVAD allows for patients with severe advanced heart failure to have the blood or the heart be bypassed in the circulatory system. So the device is implanted and the heart actually doesn't beat any longer, and the blood just flows through the device and is better perfused through the tissue by the device than the heart would be originally. And the significance of the device is that we have patients who respond really well and patients who respond really poorly, and it's really hard to predict how a patient's going to respond. And one of our goals with our work with this protein is to identify a patient population that doesn't do so well compared to those who do, and use it as a prognostic technique for identifying who should get these devices and who shouldn't. The surgical implantation is extremely expensive, and it's very invasive and difficult to go through a procedure like this, especially in a vulnerable patient population that already has end-stage heart failure. So this is really very unique system and can be really complex to talk about. In my slides, there's also going to be some papers that are cited. If you're interested in it, I'm happy to talk to you more about it, and you can also read the papers that have been cited on the slides that I provided you. But essentially, we are running ELISA kits on all the samples, which is a way to capture the protein, and then we run it through a colorimeter and measure the concentration of the protein that's left over after we've analyzed it, and then we'll model the response of the protein over time. So we've taken before surgery, 1 month, 3 months, and 6 months after surgery, so 4 different time points for our patient population, and we'll model the change of the protein over time. So yeah, these are my support people. So thank you. Water?
Yes. Why not stand? You can turn this into a Mike Knight. So unfortunately, I gotta hold it, sorry. So unfortunately, I'm not gonna have any cool slides for you guys. I don't have a great, awesome presentation with words that I still don't understand, but I will try my best. So my name is Sean Wahlgren. I'm a transfer student from Clackamas Community College. I started my research career out there in a biology course that tried to get their students to create a research question and to answer that research question themselves through so your first semester is just your basic training, and then your last 2 semesters, your lab courses are design your own experiment and test your own research and figure it out, and if you fail, you fail, and if you succeed, you succeed. So I spent an entire year doing a project that ended up failing, and I experienced what failure was like and was really discouraged and thought that, research wasn't going to be for me, and, kind of questioned a lot what I was doing. And then I realized that even if you get 0 results or not the results that you were anticipating, those are still results. Those are still something that you can learn from. And even if you get to the end and you end up not being able to parse out information from your actual results, you're able to figure out what you can do better in the future, and being able to look at any kind of negative into a positive, because the amount of times that you're going to succeed in research and find exactly what you thought you were going to find is going to be very, very slim. So getting used to that is really important. So like I said, it started at CCC. My project was looking at the effects of BPA on microscopic nematode C. elegans, if you guys are familiar. Then I moved from CCC to Portland State University as an Exito scholar. I got placed into a really cool cancer biology lab. I thought it was going to be great, but then I realized I actually don't like microbiology too much. So if you're doing cancer biology and your model organism is E. coli, you're kind of, hopefully you like microbiology. So I realized that that wasn't the spot for me. But I learned some cool things along the way, and again, I learned how to accept kind of, not necessarily defeat, but accept like hurdles along the way and keep moving forward. Now today I'm a member of the trauma research team up at Oregon Health and Sciences University. My research mentor Sam spoke earlier this morning. It is an amazing fit. I absolutely love it. I go to work in the morning up on the hill. I take the tram up. Being surrounded by a bunch of people who are in positions where I hope to be one day. It's a really cool atmosphere. I wear a pager on my hip. When a trauma gets called to the hospital, they show up at the ER, I get a page, I show down to the ER. I'm standing there with the trauma team, with the surgeons, and trying to get them enrolled into certain clinical trials that the hospital has going on, and to see if they would be a good candidate for any of those. And you see amazing, cool things. Like I find them fascinating. Having a patient come in and their heart's not beating, so the only thing that for the surgeon to do is to open their chest right there, in the emergency room, and try to save this person's life. And it's amazing because these people actually save lives every single day that they go to work. It's not just there's a lot of times where emergency physicians and stuff get kind of bogged down with what they see on a day-to-day basis. People maybe taking advantage of the system and going into the hospital for reasons that they maybe shouldn't be there. But in the trauma setting, these are people that don't have any choice. They got into a horrible accident and now they're showing up, and so being able to be a part of the team that helps to save their life is absolutely amazing. What I wish I would have known is kind of just thinking really hard about what interests me and what subjects in school I'm inspired to follow outside of class, because those are the kind of areas that I wish I would have focused on initially in having those spots potentially be for my lab. Because if you have some kind of subject that really piques your interest and that you're passionate about now, if you can turn that into your career, then you can get paid for something that you love to do and you don't ever have to work a day in your life. I think that's a good gig. Some tips along the way. Make sure you guys find a good work-life balance. You're going to be working an awful lot. I hate to break it to you, but it's going to happen, right? It's going to be great for you guys, but it's going to be easy to get bogged down and easy to potentially get burnt out. So make sure you find a good work-life balance initially. That will be very successful for you guys, or helpful for you guys. And make sure you take care of yourself. Make sure you get plenty of sleep, and make sure you go to the gym and get outside so you don't have to study all this time. Make sure you break up your days and just make sure you have a healthy learning environment. You'll thank yourself. That's all I got.
Hey everyone. So my name is Diana. I work in Dr. Stedman's lab, the virus man. He studies the viruses from hell, which is absolutely fascinating. So I started off, my interest sparked when I took my first microbiology class, and I was just so absolutely fascinated by the fact that something so small, something that's 100 nanometers in length, could put our lives in dangers, us that are like, 5 or 6 feet tall, So knowing that I had this interest, I really looked forward to going to RLC that also matched this interest of mine. And here comes along Dr. Stedman, my idol, my love. He does exactly what I love, which is really amazing. And pretty much what my research does what I'm doing right now is I'm studying the effects of silver nanoparticles, which are these little tiny, also very small pieces of silver. And I studied their effects on viruses. So can these silver nanoparticles be a new source of fighting viruses? And this is really huge because vaccines are really specific to the type of virus they